Warm good afternoon to all of you. Today we are having very distinguished Dr. Sanya Kumari ma'am. She is working as a associate professor in the Department of Dairy Microbiology at SGIDT, Basu, Patna, Bihar. She has done her graduation from the TCA Dohli, RAU Pusa Bihar, and MSc and PhD microbiology from CCS CHAU Hisar, Haryana. She has joined as an assistant professor cum junior scientist in the Department of Microbiology in Rajendra Agriculture University in 2006, and so there for 4 years. Then thereafter establishment of Bihar Agriculture University, she serves there for the another 3 years. At present, she is working in the dairy microbiology, Sanjay Gandhi Institute of Dairy Technology since last 11 year. In total, she has a total working experience of 18 years and now expertise in the field of dairy microbiology too. She served the department as a head for more than 4 years. She has carried many research projects like lychee wine as a PI, soil biodiversity and biofertilizer as a co-PI, in establishment of crop specific biofertilizer unit for the improvement of production in the agroclimatic zone 3 under RQI as a PI, and microbial evaluation of NIRA, that is government under the government of Bihar as an
Bihar.
associate scientist. She have more than 50 publications in the reputed journals like Indian Journal of Microbiology, Agroecology, and others. She has also authored the 5 book chapters and many
Chapters.
articles in the popular publications. She also authored the book on microbiology. She is the content creator of Fundamentals of Microbiology on ICR e-learning portal, that is NAHEB Comp 2, with the rating of 5. She has been awarded as a Best Teacher Award in college and university level. Also, she has the prestigious awards like Best Teacher Award, Young Scientist Award, Best Researcher Award. She is a reviewer and member of editor board of ARCC and professional journals. At present, she is handling one project on the optimization of process for the development of sweet potato enriched yogurt, and also she has filed a patent for another value added yogurt. So friends, today she will deliver a lecture on the role of fermentation technology in sustainable food production. So ma'am, I welcome to you. You can start your presentation now.
Thank you, sir. Thank you very much. And since I am expertise in fermentation, so my topic is role of fermentation for sustainable production. As your topic tells that the recent advances in agricultural technology for sustainable development of agriculture, veterinary, fisheries, and allied sectors. So I will let you know the role of fermentation, how this fermentation will help to obtain the sustainability in the various products like agricultural and other products. So friends, as we know that the fermentation is a reduction reaction in which one product is oxidized and another product is reduced, and this is basically it's found in the human being as well as in the microorganisms as also. The microorganism use this fermentation for the production of many substances. As for as if we consider the dairy product, then there is two type of fermentation, like lactic acid fermentation and there is an alcoholic fermentation. So again, lactic acid fermentation is two type: heterofermentative and homofermentative. You might be knowing that the homofermentative lactic acid fermentation in only lactic acid, and it is useful for the production of yogurt, curd, and kefir, kumis, and another microbial product. Likewise, this alcoholic fermentation used in the beer and wine production, various alcoholic products, and many other milk product also. So as we know, what is fermentation technology? Fermentation is a metabolic process that produces chemical changes in organic substance through the actions of enzyme. Industrial production of fermented product involves selection of efficient microbial strains, their culturing and preparation of suitable media, and optimization of fermentation parameters like temperature, pH, aeration, agitation, etc., and their mass production and further downstream processing like that is purifications. Now, this is the overview of fermentation. That fermentation is basically it's involved the pyruvate as an intermediate product, and that pyruvate undergo lactic acid fermentation and alcoholic fermentation. So lactic acid fermentation in many products like soy sauce, cheese, yogurt, and that involve many microbial agents like aspergillus and lactobacillus. Likewise, another important microorganism, Saccharomyces cerevisiae, it is involved in the ethanol production and like bread production also. So these are the various key ingredients for the fermentation process. Like we should have first the production strains, then we should have a proper production media and other parameters. For the production strains, you must have the high yielding strain of fermentative microorganisms. The fermentative microorganism must yield the stable biochemical characteristics, and that fermentation should not produce any undesirable substance other than your what you desire. You want to produce the substances, and that can be easily cultivated on the large scale production for the maximum yield. Likewise, the production media should have the suitable chemical composition. The precursor must be cheap and easily available. It should have the buffering capacity so that any changes in pH can be adjusted by this buffering capacity ability. There should be the ferment production media must be anti-foaming, because this foaming ultimately retard the production of fermentation, and it must be free from any toxic substances and any other microbial contaminations. The parameters include there should be optimum temperature for every type of fermentation. There is a temperature. There may be two type temperature. The temperature may be psychrophilic. The temperature may be mesophilic. The temperature may be thermophilic. As you all know that the psychrophilic temperature involve low temperature, 0 to 7 degrees centigrade. Mesophilic temperature involved 25 to 35 degrees centigrade, and thermophilic temperature is more than 45 degrees centigrade. So whatever the product, whatever the condition is there, whatever the product required, the temperature we should have to choose the optimum temperature, and the microorganism that work on that particular temperature. pH should be optimum for any kind of microbial processes. pH, there is various pH, like the microorganism like bacteria require other type of pH, other pH, fungi require other pH, and other, if we use yeast and mold, that have another pH level to show its maximum and optimum ability. Oxygen availability should be there, and water activity is there. And you know the microbial activity must require the water activity, available water activity. So there is certain optimum moisture requirement for the activity of microorganisms. So these are the various key ingredients for the fermentation process. Now there are certain fermentation products that involve various kind of microorganisms, like Swiss cheese. As we all know, the Swiss cheese is having eye-like structure, a hole-like structure, and it involves propionibacteria, and that propionibacteria is able to produce carbon dioxide, and due to this carbon dioxide, Swiss cheese look like the whole structure, that the eye-like structure appear in the Swiss cheese. The another product is cheddar cheese or yogurt that involve aspergillus, lactobacillus, and streptococcus species. Another product, beer and wine, that involves Saccharomyces species. The Saccharomyces is commonly known as yeast, and it produces carbon dioxide and ethanol. In vinegar production, again, Escherichia coli and Acetobacter is required, and for many sterilizing agent like acetone and isopropyl and clostridium species is required. So these are the various organisms and their fermented products. A wide range of product can be obtained from the fermentation, and some of the product which is industrially important, like antibiotic, organic acids, enzymes, vitamins, amino acids, solvents, single cell protein, microbial insecticides, and fermented foods. So friends, as we all know that there's in market there's penicillin, streptomycin, tetracycline, and glycofulvin. These all antibiotics are very important for today's scenario, as we are facing so many diseases. So this antibiotic, again, we obtain from the fermentation process. Organic acids like citric acid, lactic acid, acetic acid, and gluconic acid is also we can obtain from fermentation process. We also obtain this product from fermentation process, and enzymes like amylase, proteases, and pectinases. Again, these processes require fermentation process, like vitamin B12, B2, vitamin C. This process also required fermentation processes. Amino acids and solvent like ethyl alcohol, glycerol, acetone, butanol, butanediol, etc. And as we all know, we are having dosa, idli, and other fermented foods, and we know the advantage of fermented products, that it is easily digestible, and since during digestion the higher molecules is broken down into the small digestible molecules. So this fermented food is highly beneficial for our gut, highly beneficial for our intestines. So now the first product is antibiotic, and antibiotic is nothing, any chemical compound that acting against any life is called the antibiotic. And this antibiotic can be produced by the subculture culture methods. And there are two types of antibiotic, like semi-synthetic, that is ampicillin and methicillin, in which these products are obtained as a semi-synthetic, and again the further processing is required for its complete bioactive products. Another is synthetic nalidixic acids. Another type of antibiotic based on the structure is like penicillium and other related antibiotic that based on the beta-lactam structure. Aminoglycosides structure is found in the streptomycin. Macrolide structure are found in the erythromycin. Tetracyclines in the chlorotetracycline, peptides in the bacitracin, and antifungal is found in the this clotrimazole is used as an antifungal agent, and other is nitrobenzene derivatives. The various strains used for the this production of antibiotic is like cephalosporin obtained from Acremonium chrysogenum. Giladinamycin is obtained from Streptomyces hygroscopicus. Erythromycin obtained from the Streptomyces erythus. Streptomycin obtained from the Erythromyces griseus. Tetracycline from Streptomyces aureofaciens. Vancomycin from the Streptomycin orientalis. So these are the various antibiotics, and some of the antibiotic I listed here, and the associated microorganisms is just I cited few of them. Now, how this penicillin can be obtained? As you know, this penicillin is the first antibiotic ever used for the treatment of many diseases, and it is firstly discovered by the Alexander Fleming. And it was nothing. It was a chance discovery. Alexander Fleming was just plating some another fungi, and he saw that certain fungi is able to inhibit the activity of streptomycin. So the penicillium is obtained from the species Penicillium chrysogenum. Alexander Fleming used at that time Penicillium notatum. So precursor is phenylacetic acid, and for penicillin, and typical the production media contain corn starch, liquid, lactose, glucose, calcium carbonates, potassium dihydrogenate phosphate, edible oil, and other products. And it is just this is the flow diagram of fermentation process, how we just obtain the preserved culture, and we just scallop that preserved culture from mother culture to this freeze-dried spores to the agar-slant culture, that is the mother culture, and then the intermediate culture, and again the production culture. So you can see the scaling of this product from this freeze-dried spores to the this production stage. We just started from 1 liter to 1 lakh liter production stage. So these are the various fermenter and the attachment of the fermenter, like there is a arrangement of oxygen, there is agitation, there is arrangement of feeding of nutrient, and again the downstreaming process. So these are the just overview of penicillin production. Now another inhibitory substance is obtained from Bacillus licheniformis, and this Bacillus licheniformis is isolated from the goat milk, and it have the molecular weight like 18,9.19 Dalton, and it have the antimicrobial activity against many of the microorganisms like Klebsiella rhizophila, Salmonella typhi, Listeria monocytogenes, Salmonella paratyphi, Streptomyces aureus, Klebsiella, and Aspergillus hydrophila. So you can see the zone of inhibition against the Streptococcus Salmonella typhi. It's not a Streptococcus, it's a Salmonella typhi, which is a pathogenic of it causes typhoid in human being. So we can obtain this antibiotic from Bacilli licheniformis by the process of fermentation. On the mode of action, it inhibits the RNA biosynthesis in their target organism, and thereby the skill just stop their growth. So another microorganism is Bacillus brevi, and this can be isolated from the infant feces sample, and its high molecular weight, it is 2,367 Dalton protein, and it have again antimicrobial activity against Klebsiella rhizophila, Listeria monocytogenes, Streptococcus aureus, and Mycobacterium smegmatis. Again, it stopped the synthesis of RNA and thereby kill the target microorganisms. So many organic acids can be obtained from the fermentation process, like acetic acid, formic, propionic, citric acid, succinic, lactic acid, and you all know how these organic acid are useful nowadays, and like we can have citric acid from Aspergillus niger, Candida lipolytica, lactic acid from Lactobacillus delbrueckii, acetic acid from Acetobacter aceti, gluconic from Aspergillus niger, pyruvic acid from Pseudomonas aeruginosa, succinic acid and tartaric acid from Bacterium and Bacterium succinum, and Gluconobacter suboxydans respectively. So how can we obtain the citric acid by the fermentation process? The organism used is Aspergillus niger, and again it is a surface culture or submerged culture process. So media composition is the composition of media is sucrose, ammonium nitrate, potassium dihydrogen phosphate, magnesium sulfate, heptahydrate, hydrochloric acid, etc., etc. And as we know that there is wide use of citric acid. The citric acid used as a preservative in the jam and jelly. Citric acid is used as a precursor for the many medicines. So it is very useful product for the pharmaceutical industry. It acts as a chelating and sequencing agent. So it can be used in the tanning of the animal skins, and citric acid nowadays is used in the various carbonated beverages. So this is the flow diagram for the production of citric acid. So we obtain the Aspergillus niger. We can have various strain. So Aspergillus niger SM101 is used here, and that was subculture on the PDA slant for 7 days at mesophilic temperature, and that inoculum was developed in the spore was collected, and it was suspended in the 0.9% sodium chloride, and it was inoculated initially from in a 250 ml flask, and containing and that was again agitated at 200 revolution per minute. And at that time, addition of ethanol and ethanol is done, and for their activity, there's some wheat molasses is added. Then filtration, the supernatant for citric acid estimation, the supernatant contains citric acid, and residue contain the cell biomass, and that cell biomass can be recycled or can be reused for the production of citric acid. There are many microbial enzymes that are used for the various detergent industry, and as you know, that enzymes are protein that help any metabolic activity or any chemical reactions inside or outside our body. Inside our body, it's an enzyme, and outside our body, it is catalyst. So this enzyme is very critical for our metabolism. So most industrial enzymes are produced by the microorganism, and in these enzymes, mostly fungi and bacteria are grown under a well-defined condition in either submerged or in the solid state fermentation. There are various types of microbial enzyme like amylase that can be obtained from the Aspergillus oryza, Aspergillus niger, glucoamylase from Aspergillus niger, and Rhizopus nevaeus. Proteases that is used in the detergent industry, it can be obtained from the Aspergillus, lipase from the Candida cylindrica, cellulase from Trichoderma viride, aspartase, catalase, aspartase from E. coli, and catalase from Aspergillus niger. So for this amylase production, Aspergillus niger and Aspergillus oryzae or Bacillus lecciniformis can be used, and amylase biosynthesis is inhibited when the medium contains glucose. So the medium composition must be devoid of any glucose material. So medium composition contain cornstarch, cornstick liquor, potassium chloride, calcium chloride, magnesium dichloride, hexohydrate, sodium hydrogen phosphate, etc. And wheat bran is commonly used for the raw material. So here you can see the Aspergillus. Aspergillus is commonly a black mold. Now, there are the tremendous use of enzyme. Enzyme can be used in the detergent industry. Enzyme can be used for the starch or saccharification or liquefaction industry. This enzyme can be used in the paper and textile industry. It can be used for the bioremediation, for anti-biofilm activity, wastewater treatment, de-emulsifying agent, and bioremediation of phenylkane, and bioethanol production, brewing, and bakery industry. So the entire industry is based upon this application of enzyme and activity of enzymes. Besides enzymes, some amino acids are also can also be obtained from the fermentation process. So amino acids are important class of compound, and that required by the living cell as a growth factor. The production of amino acid to fermentation was firstly initiated by the Kimosita and his co-worker in 1956. And various enzymes, various amino acids are there. So some of the enzyme like Corynebacterium glutamicum is able to produce L-lysine, L-leucine, homoserine, proline, histidine. E. coli is able to produce threonine. Serratia marcinus is able to produce the isoleucine. Bacillus subtilis is obtained, can be used for the arginine. So these are the various enzymes, and we all know that the enzyme is the some of the enzyme are essential and some are the non-essential enzymes. So the essential as well as non-essential type of enzymes can be produced by this by the add of microorganism in the process of fermentation. Now we can use the production. We can see the production of L-glutamic acid, and for this organism is used as Corynebacterium glutamicum. Glucose and starch hydrolysate are the principal raw material for the fermentation process, and fermentation is carried out at 30 degrees centigrade, and it is carried out for the 40 to 48 hours with the pH of 7 to 8. Media composition contain glucose, constant liquid, enzymatic casein hydrolysate, dipotassium phosphate, magnesium sulfate, and urea. And this glutamic acid is used as a flavor enhancer, and it can be it is also used in the cosmetic industry like it is used as an anti-wrinkle agent in many of the face cream, and it is glutamic acid also used in the hair restorer. So, and in animal feed, it is used as a nutrient supplement also. So, another product that we can have by the fermentation process is vitamin. So vitamin are the important compounds in diet which is synthesized by the phototropic microbes in amount to fulfill its need. But few microorganisms excrete vitamins and greatly in excess of their metabolic needs. So if we grow in a highly specified and artificial condition, we can have the sufficient number of vitamins. So there are two types of vitamin. One is the fat soluble and water insoluble. As you all know that vitamin A, D, E, and K is the fat soluble, and other than these enzyme, most of the enzyme are the water soluble enzyme. So again, vitamin production involves the fermentation process. Here, the Bacillus megaterium, Propionibacterium, many Bacillus coagulans, and Pseudomonas nitrificans microorganisms can be used for the production of vitamin B12. And this vitamin B12 is also known as the cyanocobalamin. And daily requirement of this vitamin is 0.001 milligram per day, and it is manufactured by the submerged culture. And with the process of aeration and agitation, so this vitamin B12 is used as an antidote for the cyanide poisoning in the treatment of Persinius anemia, responsible for reducing brain shrinkage, and it is very helpful in the healthy regulation of the homocysteine controls. So these are the diagram for the downstream fermentation and upstream process. In the upstream process, we just add the nutrient requirement of that microorganisms, water, and then many attachments are there for the fermentation. Then fermentation, the pH control, aeration, agitation system is there, and downstream processing, that is the product recovery, and cell separation and crystallization of the products, and spray drying for to be used as a powder or other form to be used in the now. So we can conclude that the microorganism is a very useful, and it has very high potentiality to produce an array of metabolites, and that products can be used to obtain the sustainability in our production. So it's a many health benefit products that like enzymes, anti-inflammatory, act as anti-inflammatory agent, anti-arthritis, anti-diabetes, anti-hypercholesterolemia properties, etc. nowadays. And microbial diversity must be explored for the obtaining newer substance from the nature. So thank you very much. So now,
Any questions from participants?
I'm not able to see any participants.
Okay, ma'am. There is one question. I will read for you.
Yeah. How do we come to know? Yes. How do we come to know that the fermentation product produced after fermentation is enzyme, antibiotic, or any other compound with some other role? See, for every for every product, for every fermentation process, we must have specific microorganism, specific growth requirement, specific substrate. So if we need enzyme, if we want to produce enzyme, then some specific microorganism will be there, the substrate will be there. So the process is entirely different for the enzyme and the antibiotic. So you know what you are going to produce. You know, if you are going to produce enzyme, then you know you should add that type of microorganism, that type of precursor, that type of fermentation condition. Specific media is there. So we know that if we are going to produce enzyme, we will have enzyme. If we're going to produce antibiotic, then we will have antibiotics. Enzyme have another property and antibiotic have another property. So there will be different growth requirement for the both microorganism, for both the product, and the substrate will be different for both the product. Enzyme for enzyme, another fermentation condition will be there. For antibiotic, another fermentation condition will be there. Substrate will be different for enzyme. Substrate will be different from the antibiotic for the antibiotics, and the microorganism will be different for enzyme and different for the antibiotic also. Is it clear? Hello? Parjade Zubair Ahmed? Okay, thanks. Next, please. Acha, can I ask some question from you? What is lactic acid fermentation and glucose fermentation? How will you differentiate between these two fermentation? Anybody? Anybody else? Hello? ये मेरी आवाज नहीं जा रही क्या? Hello? हेलो सर.
मैम आ रही है मैम.
अच्छा, तो दूसरे भी क्वेश्चन पूछे प्लीज.
ये अभी सर मैडम आपने डिफरेंस बोलने के लिए बोला
हाँ
क्या?
पूछा, किसी ने कुछ बताया नहीं मुझे.
तो lactic acid fermentation is a metabolic process ना, by which glucose and other 6 carbon
Metabolic
sugars
process.
are
Both are
converted.
the metabolic process that happen in our body also. Glucose fermentation.
And it
Glucose.
will be converted into cellular energy ना.
See, glucose fermentation is the glycolysis and lactic acid fermentation. So lactic acid fermentation, it's happen in our muscles basically. When we do exercise, then lactic acid produced in our muscles, and it creates some pain due to the production of lactic acid. And in glycolysis, glucose is converted into pyruvate, and ultimately it is whether it converted into alcohol, or if this glucose entered into the TCA cycle, tricarboxylic acid cycle, then it entirely converted into carbon dioxide and water. But the basic difference between this glycolysis and lactic acid is that in glycolysis we obtain 4 ATP, and in lactic acid we don't have any ATP production. There is no ATP production in the lactic acid.
Yes. Thank you, ma'am. Thank you.
Fatigue, okay. 2 ATP. Oh, yes. 2 ATP, and in glycolysis 4 ATP is there. But so ask me another question related with fermentation or any other product. What is the difference between, have you heard about the yogurt or curd? Can you differentiate between the curd and yogurt? Hello? See, we talk about the sustainability in production now, so we should have an array of product to meet our daily requirement. So yogurt, curd, and other whatever the dairy microbial product is there, that will ultimately meet the sustainability, that will ultimately fetch the farmers income. So, do you know curd, yogurt? Have you heard about this two product? But is there? I think they have just joined and doing some other work. But okay. You don't know the difference between. Please answer. So, curd, you know? Curd,
Yes,
you know?
yes,
Curd,
ma'am. Yes.
yes, curd, you know, and yogurt, you don't know.
No, yogurt,
Markets, yes,
madam,
yogurt bhi aap
ma'am.
jante ho.
That some flavored yogurt is available in the market. I think in jamun flavor, in strawberry flavor, or rose flavor.
हाँ, ये फ्लेवर्ड भी होगा, नॉन फ्लेवर्ड भी होगा. कर्ड क्या होता है, बेसिकली ये इंडियन प्रोडक्ट है जो कि हर घर में बनाया जाता है. और कर्ड में हम लोग क्या करते हैं, मतलब हमारा कुछ पता नहीं रहता कि उसमें क्या माइक्रो ऑर्गेनिज्म, वी डोंट नो द कंपोजिशन ऑफ माइक्रो ऑर्गेनिज्म. वी सिंपली ऐड सम इनोकुला फ्रॉम द प्रीवियस lot of curd, and then we add in the warm milk. So there is no, we don't know the microbial flora in the curd, right? But in yogurt, selected microorganism is there. Yogurt culture is there. There's so many type of yogurt culture. Some use bifidobacterium, some use lactobacillus, some use streptococcus. So we know the exact composition of yogurt. The microbial composition will be fixed, whether they will be in single culture or will be in the double or triple culture, and the fixed inocula percentage will be there. But in curd, there is no you can't say that these are the composition of curd microflora. So this is the basic difference between the curd and the कर्ड इज क्या, मतलब घर में जैसे बनाते हैं वो कोई उसका कोई कंपोजिशन हमें नहीं पता, वो एक कंट्रोल कंडीशन में नहीं होता. बट योगर्ट में हमें माइक्रोबियल कंपोजिशन पता चलता है, उसका जो टाइम होता है वो हमें पता चलता है कि 4 घंटे में उसको जमना है तो 4 घंटे में हम योगर्ट ले सकते हैं. उसका जो एक टेम्परेचर होता है वो एक ऑप्टिमम टेम्परेचर होता है, पीएच एक ऑप्टिमम पीएच होता है. सो हम जब ट्रेडिशन के जब घर की बात करेंगे तो वो कर्ड हो जाएगा, जब हम उसको इंडस्ट्रियल लेवल पे ले जाएंगे दैट बिकम द योगर्ट. और योगर्ट फ्लेवर्ड भी होता है, कई तरह के उसमें हम लोग मिलाते हैं, फ्रूट्स वगैरह हम ऐड कर सकते हैं, सीरियल भी आजकल एडिट किया जाता है. सो फ्लेवर्ड भी होता है और एक प्लेन भी होता है, कई तरह के योगर्ट होते हैं.
Ma'am, nutritional point of view, which is better, yogurt or curd?
In nutrition, from nutritional point of view, both is good. बट जैसे कर्ड में क्या होता है ना, हमें पता नहीं चलता है कि उसका सेल्फ लाइफ क्या, हम खाते जाते हैं. जैसे अगर हमने फ्रिज में रख दिया 2-3 दिन के बाद उसकी एसिडिटी बढ़ी घटी, वो हमें नहीं पता चलता क्या, मतलब फ्रिज में क्या उसमें कुछ माइक्रोबियल ऑर्गेनिज्म भी ऐड हुए हैं बाहर से भी, कुछ इस टाइप का हमें नहीं पता चलता. बट योगर्ट क्योंकि इंडस्ट्रियल लेवल पे होता है, उसकी टाइम टू टाइम जो है ना वो सेल्फ लाइफ आपको उसमें दिया रहेगा कि आप इसके बाद उसको नहीं खाएं, अदरवाइज इसमें जो है वो कंटामिनेशन के चांसेस होंगे. दोनों जो है वो हेल्थ के लिए बहुत ही अच्छा होता है. हमारे गट में जो भी ये प्रोबायोटिक माइक्रो ऑर्गेनिजम्स के कैटेगरी में आते हैं, जो भी कर्ड के कंपोजिशन होते हैं या योगर्ट के कंपोजिशन होते हैं, वो प्रोबायोटिक माइक्रो ऑर्गेनिज्म के कंपोजिशन में आते हैं. और प्रोबायोटिक माइक्रो ऑर्गेनिज्म वो होता है जो हमारे गट हेल्थ को बहुत अच्छा रखता है. मतलब उससे हमें जो हमारा इंटेस्टाइन होता है उसमें कई तरह का ये एंजाइम्स एंड कई तरह के ये ऑर्गेनिक एसिड का सेक्रेशन करता है, जिससे कि हमारा हेल्थ जो है वो अच्छा रहता है और हमारा जो बॉवेल मूवमेंट या कई तरह के जो भी और भी जो गट रिलेटेड प्रॉब्लम है वो दूर हो सकते हैं. सो दोनों ही इज इट खोआ फर्मेंटेड प्रोडक्ट? नो, खोआ इज नॉट फर्मेंटेड प्रोडक्ट. खोआ को तो हम लोग क्या करते हैं, उसको कंडेंस करते हैं. खोआ को आप क्या करते हो, वाटर उसमें से रिमूव करके और उसको गाढ़ा बनाते हैं. तो खोआ इज नॉट फर्मेंटेड प्रोडक्ट. Many farmers are preparing homemade rice solution in a northern polyester instead of humic acid. How does it work, the humic acid? When you are preparing homemade rice solution, homemade instead of humic acid, how does it work as a humic acid? मैं क्वेश्चन नहीं समझी. देखो, राइस होममेड राइस सोल्यूशन अर्दन पोर, तो बिटवीन कर्ड एंड योगर्ट, देखो कर्ड एंड योगर्ट के फैट परसेंटेज में डिपेंड कर, बिल्कुल ही डिफरेंस रहेगा। आप किसका मिल्क यूज कर रहे हो, वेदर आप स्टैंडर्ड मिल्क यूज कर रहे हो या फुल फैट क्रीम यूज कर रहे हो, मतलब फुल फैट मिल्क यूज कर रहे हो, या आप उसमें से मतलब एडिशनल कुछ उसमें फैट डाल रहे हो, तो अब मिल्क के कंपोजिशन पे फैट का कंपोजिशन जो होगा वो चेंज करेगा। अगर आप कर्ड में भी घर में आप किस तरह का फुल फैट क्रीम यूज कर रहे हो या विदाउट फैट क्रीम यूज कर रहे हो, तो उसका फैट वेरी करेगा। योगर्ट का भी सेम है, वो डिपेंड करता है कि आपने कैसा मिल्क लिया है, बफेलो लिया है, काउ लिया है, उस पर डिपेंड करेगा फैट परसेंट। एनी अदर क्वेश्चंस? ये क्वेश्चन में नहीं समझी आपके, ये जो ना होममेड राइस सोल्यूशन इंस्टेड ऑफ ह्यूमिक एसिड। ह्यूमिक एसिड अलग चीज है। आप ह्यूमिक एसिड क्या है, वो मतलब वो एक अलग चीज के बारे में बात कर रहे हो। वो तो हो गया आपका कंपोस्ट वगैरह में जो है ना, ह्यूमिफिकेशन के थ्रू ह्यूमिक एसिड वगैरह बनता है। और राइस सोल्यूशन को जब आप फर्मेंट करोगे तो उसके अलग प्रोडक्ट्स बनेंगे। राइस से कई तरह के बिवरेजेस भी बनते हैं। तो ये क्वेश्चन थोड़ा सा समझ नहीं पा रही मैं। टेल अबाउट कैफीन, देखो कैफीन एंड क्यूमिस ये ना मतलब अल्कोहलिक प्रोडक्ट होते हैं, मिल्क से अल्कोहलिक होते हैं, मतलब कि समवट इसमें अल्कोहल होता है। इसमें आपको लैक्टिक एसिड भी यूज होता है और इसमें कुछ ईस्ट भी मिलाया जाता है, ठीक है ना। कैफीन एक मतलब इंडिया में तो इतना पॉपुलर नहीं है, फॉरेन कंट्री में ये ज्यादा कैफीन एक ड्रिंक होता है और इसको मिल्क से ही बनाया जाता है। पहले ये ना कैमल मिल्क से बनाया जाता था, बट अब नाउ ए डे इसको काऊ के मिल्क से भी बनाया जाता है। और इसमें बस यही है डिफरेंस कि ये ना इस हल्का अल्कोहलिक रहेगा। कैफीर में अल्कोहल का परसेंट ज्यादा, दो प्रोडक्ट है कैफीर एंड क्यूमिस। क्यूमिस कैफीर में तुम्हारा अल्कोहल का परसेंटेज थोड़ा ज्यादा होगा, लैक्टिक एसिड भी होगा एंड अल्कोहल भी होगा। एंड क्यूमिस में क्या है, अल्कोहल का परसेंटेज आपका कम रहेगा, थोड़ा कम रहेगा। और दोनों के जो माइक्रोबियल कंपोजिशन है ना, जो उनमें जो माइक्रो ऑर्गेनिज्म डाला जाता है उसके भी कंपोजिशन में थोड़ा बहुत ये रहता है, बट बेसिकली लैक्टिक एसिड एंड जैसे सैक्रोमाइसिस, कैंडिडा, ईस्ट भी यूज किया जाता है, कुछ फंजाई एंड कुछ बैक्टीरिया, ये दो यूज होते हैं।
Ma'am, it means that we are able to prepare the wines. So you are putting the examples of Saccharomyces, na? So can it possible to prepare the wine from milk?
देखो वाइन बनाने के लिए ना आपको अल्कोहल का कोई भी चीज जो आप ड्रिंक करते हैं ना, जैसे बियर का अल्कोहल परसेंटेज होता है, वाइन का अल्कोहल परसेंटेज होता है, फिर आप ऊपर जाते हो शैंपेन जो भी है, वोडका वगैरह, सब ये जो प्रोडक्ट बने हैं ना, ये वाइन ये अल्कोहल के कंपोजिशन से बने हैं। मतलब अल्कोहल का कितना परसेंटेज जो है वो बढ़ता गया, जैसे बियर में थोड़ा कम होता है, फिर आप ऊपर जाओगे वाइन में तो उसमें थोड़ा सा और ज्यादा रहेगा, मतलब इस तरह का होता है, राइट। जो मिल्क में ना बहुत स्लाइट अमाउंट ऑफ अल्कोहल होता है, बहुत ज्यादा अल्कोहल नहीं होता है उसमें। तो आप मिल्क से वाइन नहीं बना सकते। वाइन बनाने के लिए आपको फ्रूट ही जूस यूज करना मतलब फ्रूट ही कंपोजिशन आपको, क्योंकि वो जो मतलब कि जो फ्रुक्टोज पे जो बैक्टीरिया जो माइक्रो ऑर्गेनिज्म है वो फ्रुक्टोज पे काम कर सकता है करता है। यहाँ पे आपको लैक्टोज है, फ्रुक्टोज नहीं है, ठीक है। तो फ्रुक्टोज और लैक्टोज की बात करेंगे ना, तो फ्रुक्टोज जो है वो इजिली अवेलेबल है और फ्रुक्टोज जो है वो बहुत जल्दी जो है वो चेंज हो जाता है इन अल्कोहल में। बट लैक्टोज में उतनी कैपेबिलिटी नहीं रहती कि वो अल्कोहल में चेंज करे। हमें दूसरा माइक्रो ऑर्गेनिज्म डाला जाता है, बट वो भी एक कंट्रोल मतलब कि उसमें डाला जाता है। कैफीर में अल्कोहल का परसेंटेज स्लाइट रहता है, बहुत ज्यादा नहीं रहता है, अल्कोहलिक है बट इट इज स्लाइटली, बहुत कम अमाउंट होता है उसमें अल्कोहल का। So for beer,
Thank you, madam.
I have sent the YouTube video for your reference. Okay. For what caffeine? YD Paramar. अच्छा फार्मर, अच्छा ह्यूमिक एसिड हाउ डज इट वर्क एज ह्यूमिक। मैं एक्चुअली क्वेश्चन आप मुझसे थोड़ा सा टॉक करें ना तो मैं थोड़ा सा समझ पाऊँ। YD पावर जी, many farmers are preparing homemade rice solution in an earthen pot instead
हेलो।
of ह्यूमिक एसिड। हाँ जी,
हाँ मैडम, मैडम
हाँ
वो जो मैंने वीडियो भेजा था, मतलब फार्मर्स ऐसे प्रैक्टिस एडॉप्ट कर रहे हैं, तो उसके रिगार्डिंग मैडम क्या वो कैसे हाउ इट वर्क्स एज ए ह्यूमिक एसिड दैट सॉल्यूशन?
देखो बहुत ज्यादा तो मुझे नहीं पता है ये ह्यूमिक एसिड कैसे, मैं सिर्फ थोड़ा बहुत कंपोस्ट के बारे में बता सकती हूँ, है ना।
ओके
मतलब जैसे राइस सोल्यूशन को वो एज ए क्या यूज कर रहे हैं, एज ए फर्टिलाइजर और एज ए
यस यस मैम
इंसेक्टिसाइड,
एज ए
एज ए
फर्टिलाइजर
फर्टिलाइजर,
ह्यूमिक
एज ए
यस
फर्टिलाइजर यूज कर रहे हैं। ओके, तो राइस सोल्यूशन को वो अर्दन पोर्ट में यूज करके एज ए फर्टिलाइजर यूज कर रहे हैं
यस मैम।
इंस्टेड ऑफ ह्यूमिक एसिड। ह्यूमिक एसिड फर्टिलाइजर है।
मतलब मैडम रूट इनिशिएशन व्हाइट रूट्स के लिए वो काम करता है एक्चुअली।
अच्छा, मैं नहीं बता सकती इसका, आई एम सॉरी।
No, ma'am. Humic
मुझे
acid,
थोड़ा
the edible grade of humic acid is not available in the market. It is only used for the sake of improve the soil health and plant health.
क्योंकि मैं बहुत ज्यादा नहीं जानती हूँ ह्यूमिक एसिड के बारे में,
ओके ओके
है ना। हाँ, मुझे बस इतना पता है कंपोस्ट, क्योंकि कंपोस्ट पे मैंने काम किया हुआ है, मेरा रिसर्च भी रहा है, तो मुझे उस पे पता है। मैंने एस्परजिलस वगैरह को ये करके ना उससे कंपोस्टिंग कराई थी,
ओके
तो उसके बारे में मुझे। चलिए और क्वेश्चन प्लीज। आप लोगों का, मैं, आप कहाँ से हैं वाईडी पवार जी?
मैडम मैं गुजरात से।
गुजरात से, एज असिस्टेंट प्रोफेसर हैं आप?
यस मैम यस मैम।
ओके, नाइस टू मीट यू। एंड बालचंद्रन जी, बालचंद्र वी, रमनदीप, डॉक्टर, अभी इनसे मंदर खान से तो मुझे मेरी बात हुई शायद, है ना। आप लोग अपना इंट्रोडक्शन देना चाहेंगे? मुझे अच्छा लगता है क्योंकि मैं भी तो फैकल्टी, आप फैकल्टी, मैं भी फैकल्टी हूँ, तो मुझे अच्छा लगेगा फैकल्टी से कुछ आपसे मैं भी जानूं, आप लोगों का फील्ड क्या है, फील्ड ऑफ स्पेशलाइजेशन क्या है ना। और कोई क्वेश्चंस है फर्मेंटेशन से रिलेटेड, मिल्क, योगर्ट, इनसे रिलेटेड कुछ भी? मैंने वाइन पे भी काम किया है, कुछ क्वेश्चन वाइन से अगर आपके हो तो। लीची वाइन पे मैंने काम किया हुआ, लीची वाइन बनाया था, बट बिहार में जो लिक्विड बैन हो गया तो इसलिए मेरा प्रोजेक्ट बंद हो गया। अदरवाइज मैंने किया था। अभी मैंने एक प्रोडक्ट जो है वो पेटेंट फाइल किया है, और जो वैल्यू एडेड प्रोडक्ट पे, योगर्ट पे ही मैं काम कर रही हूँ अभी राइट नाउ, बिकॉज आई एम इन द डेयरी माइक्रोबायोलॉजी, है ना। और इसके पहले मैंने बायो फर्टिलाइजर पे भी काम किया है, दैट इज अनदर ब्रांच ऑफ फर्मेंटेशन। सो और कुछ पूछना हो आपको फर्मेंटेशन से रिलेटेड, या जो भी मैंने फील्ड आपको बताए?
Any questions? I think, ma'am, there are no more questions.
Okay, so
Ma'am, it was really a nice lecture, very interesting. Thank you so
Thank you.
much.
Thank you,
on
ma'am.
behalf of NADCL. Thank you on behalf of participants. Thank you on behalf of organic community. We may leave now.
Please, thank you, ma'am. Thank you all. Thank you. Thank you very much.